it's indeed a problem if you would like to work with small patient amounts or with small amounts of cells. Now, what happens when we hit our cells with antibodies, they start to change their features. And if we have a second label or Y label like magnetic beads or fluorescence beads, they indeed have not anymore the features that we know from before. So that means labeling is stressful. And in stem cells, especially, we know they change their features and we cannot use them anymore for therapeutic features. So on the other hand, if you would like to learn a little more about your cells, like what is the DNA, what has changed, what are about proteins and lipids, you always need a large amount and a fresh batch to go, for example, to molecule or the protein analysis and so on. Now, the question was, is there any possibility to learn about our cells, what changes in their proteome and what changes, for example, during their development without killing them, without the endpoint analysis? And the idea was to look into Raman spectroscopy, which is well known in physics and chemistry and also in pharmacology for long years, but nobody really realized that we can use this also for living cells and for our biological questions. I know that it's usually a bugbear for all of you have heard about Raman spectroscopy because it's very complicated and you see some patterns, but you don't really know what it is. But let me show you today that it's very easy to handle tool now. And what you do is you basically look into the interaction of into the interaction of a laser light that shines into the cell. You get all the typical things like reflection, like absorption, and so on. And some of the photons that go into the cells will be jump into the chemical bondings, and you will get so-called inelastic scattering. And this is what we look at. So what you get are different typical spectra. And now just keep in mind that each of the molecules within the cell contributes to it. So it's not only confined to the surface like the antibody-based applications. Any molecule takes part into this kind of signaling. And you can use, of course, now statistical operations to help us to understand the diversity of the cells and to see what makes the difference between the two of them. So what you get are then these little clouds. Each of these points, and you will see a lot of them during the lecture, are one cell with a background information of about 600 points, and we have n-dimensional information which are brought now down using this technology for 2D demonstration so that we can understand what's happening, what is the difference between the cells that we look at. And the most important thing is what we made is to make a machine available for us biologists so that we can handle our normal cells coming directly from the incubator or even under a controlled environment. We can use petri dishes and small wells where cells can just grow in. And now what we do is we get a fingerprint of each of the cells and can analyze them using this new technology. Let me in principle show you how it works. You shine a laser, and it's important to know that we take a 785 nanometer laser, which is the optical, in the so-called optical window of the biological samples, so it's not harming the cells at all. Then we get, as soon as the molecules go into the chemical bondings, there starts vibration. Some of them will be ejected with much higher energy than they came in. This is a rare event. And there is another event which makes the cells, the photons being ejected in a higher wavelength, which means with lower energy. And that's what we look at. This is also still a very small amount of photons, one from 10 billions. That is a so-called Raman photon, but this gives enough information for us. And we get a really fingerprint of the cells where we can pinpoint each type of cells, each developmental stage of the cells, because we get different spectra. And we know that as soon as the metabolome changes, we get another signal. And the metabolome changes due to growth, due to disease, due to drugs or toxins. And let me now show, we have started a lot of applications. We can also work in solution because we can keep the cell in an optical trap. as long as we make the measurements, and as soon as we switch off the laser, the cell will fall down and still be happy. And we can work in a very high resolution, because we can select either the nucleus of cells in culture or on the cytoplasm. If we work with cells in solution, of course, we have the entire cell because they are rounded up. And we can also work in tissue sections. We can work with fixed cells. We can work with living cells. If we see differences in fixed cells, we know in living cells it's even much more pronounced, because during fixation, you lose some of information. And we have started in tumor area. We have started with stem cells, and we worked with drug screening models. And also what I'm going to tell today is about quality assurance of your cell-based therapeutics. So what we found or started off is it is always difficult to discriminate fibroblasts from stem cells, because there is no marker available that is really clear differences for the FACS or MAX analysis. If you would really know what is the difference, you have to cultivate them, which takes about 3 weeks, which is also not very convenient if you work with patient material. But if you take Raman spectra, you can see maybe by eye already that there are some differences. But of course, the computer will help you to demonstrate the difference much more. And you see fibroblasts assemble in a completely different area than the mesenchymal stem cells, which gives you kind of an idea. I can separate them just because of the reaction with this laser light. On the other hand, if you would like to define or find good donors from bad donors, what you can use are the adipose-derived stem cells, and I learned that some of them have much more potential to go towards chondric differentiation, and the others go more to osteogenic differentiation. And the question was, can we use Raman spectroscopy as a first hint? Will this be a good donor or a bad donor? So what you usually do, you take the cells from the patient, you isolate them, and then you make spectra, Raman spectroscopy, at different time points. And here after day 4 or day 3, these are the bad control, the so-called bad donors, what they are called, and these are the good donors. So some of the cells of the good donors still are at the same place as the bad donors are, but about, I think it's 60%, 70% of them have changed. So this gives us hope that we can use Raman spectroscopy to find the good donors for this kind of treatment. We also can look what makes the difference between the two kinds of donors, the good donors and the bad donors. And if we look to the spectra and we analyze them, then we can see that in this case are the lipids and the collagen, which makes the difference between the two groups. On the other hand, having a possibility to look into a small group of cells during your experiments, what kind of change they are undergoing. I learned that between necrotic or apoptotic cells, it is sometimes difficult to really have a test to analyze them. Of course, there are antibodies available, but that means you have to kill the cells to look at them. You can't really discriminate them just by bright field microscopy. But what you can do is now make spectra from each of the cell groups. So, for example, these are groups or cell groups of a sarcoma cell line. And you see when you look to early apoptosis, this is the control group, and this is the early apoptosis. This is the late apoptosis. And you get more pronounced differences if you go to the necrosis of the cells. And you can see the spectra have some certain areas which are marked here. These areas are the wave numbers that correlate with the proteins or with the DNA information that is behind that. In this case, necrosis was introduced by using temperature differences and also necrosis to make sure that we don't get interference with biochemical molecules. But you can see that they change really very drastically, and even in this group, you see the difference between the early and the late apoptosis, and here the necrosis more and more pronounced. So it takes about, let's say, 100 cells. You just test them, and you get a feeling how your culture changes and what has maybe changed during incubation and so on. The Raman signal, which was the most dominant area, was this 1,375 per centimeters. This is the physical word of how the air create the differences. And what we could see that is progression of nuclear condensation. And we also saw differences in these nucleic bands between 780 and 800 centimeters -1. And this is usually during apoptosis because their nucleosomal cleavage started. So you get information about the status of the cells. On the other hand, we were approached by the German Red Cross Center. They wanted to see, is there a possibility to make a functional test of their blood products before they give it to the patients. So usually they have empirically found out that it takes about maximum of 6-7 days for thrombocytes to be given to the patients, and for red blood cells it's about 28 or something like this. But they measure it during the time of production. There's not a real control just right before it goes to the patient. And it is a statistical analysis that they make. And we wanted to see how can Raman spectroscopy help us to understand what happens with the cell. So we take each, these are platelets, we take the single platelets, just hold it in the optical trap during analysis, and then we measure about 30, 50 cells, depending, and we did this for several days. So we just took out microliter from the blood bag, in this case under sterile conditions, and then we analyzed it in these little wells. And between day 1 and 8, we did see that the blue lines, these are from 3 donors, they all assemble at a certain area. So it's independent from the donor as well. But after this, there's a shift. So all the spectra are now shifted in this way. And we learned that this is especially due during cell decay. This is the kind of proteins or the molecules which are making the shifts between the first group and the late groups, that's the early ones and the late ones. We tried to do the same thing now with erythrocytes. Also here we kept each single cell a short time in the optical tweezers during our measurements. And then we see between day 20, up to day 21, there was no difference between the 3 donors. But then one of the donors changed. It was at day 28, where we would say this has something changed within the blood product. The other donor was after day 30, and the last one was at least 36. That means the 3 donors have different kind of blood, which makes them more or more to be held or to be kept and preserved. And on the other hand, it could be a very big help now. Just prior, you give the blood product to the patient to test, is it still functional, available or not? So this is the first experiments we have done. We now start a collaboration with these people, also from the transfusion medicine, to learn about the capability of Raman spectroscopy as a quick approach to see, is it still good or should you throw it away? On the other hand, this might also save a lot of products because we can keep them longer and don't need to throw them away as empirically studied after day 21. Also here, these are the wavelengths that we found were the dominant ones, and we know these all contribute to cell decay. And that means we could not only look into the quality, the functional function of the blood, we simultaneously can look whether there are bacteria in there, because we can, the spectra of the blood products or the erythrocytes change when there's a bacterial infection within the material. Now, completely other approach, we have a collaboration because of EU project. It's called IDEA. It's from the FP7 Health Project, where we work together with the Paracelsus Medical University in Salzburg, and they work on neurofunctional regeneration. So what they did is they had precursors or progenitor cells, and they used TGF-beta to induce growth. And they wanted to see when or how can we see from the functional, from the precursor area to the functional area. What we did is we just measured first the control group, which is that one, and we saw this is one group. Then we used the TGF-beta treated ones, and we got another population. So what we realized is, okay, there is a gap where the people have, where the cells have the same overlap, and we wanted to see first what is the difference between those cells here and those cells, And the information that we got was that the difference between the two of them is proline. So the cells that have been seen, TGF-beta, and have a different spectra, they have change in the proline activation. So then we compared those cells within the overlap with those cells that have been outside of this overlap from the TGF-beta treated cells, and we realized the same difference. So that makes us thinking, okay, the TGF-beta treated cells obviously have part of them changed, but the others didn't. So we approached them, the colleagues, and said, so that is our results. And they said, yes, this is the same what we see with patch clamp technologies, which is, of course, time-consuming. And also, we see this by immunohistochemistry, which means it's an endpoint analysis. So that means this technology can be used as a marker to follow the change of precursor cells towards functional neurons. And the same we could do using microglial cells that have been treated with different kind of drugs, in this kind lipopolysaccharides, interleukin, and also compared now with the controls. And here we could see that there are different kind of RNA activities of these different kind of states of microglial information. Another idea is to use, this I already showed. Another idea was to look into the EPO treatment of cells. The idea was to find out how EPO treatment changes mesenchymal stem cells towards fibroblasts. And also here we can see that Raman spectroscopy depicts the cells that have been changed and those that have not been changed. And in case of you would like to work with models to find out bronchial diseases, you usually take patient material. So you don't know are these tumor cells or non-tumor cells. So before you put it into your devascularized cells or the devascularized scaffolds, you should make sure that they are new tumor cells so you can discriminate between tumors and non-tumors, and there are some tumor factors, so the differences that you see here are known as tumor-relevant factors. This was also published, and it is also very important to see that between cells that have been treated with hypoxic or normoxic treatment, you see differences very soon, and cells that have been working in 3D scaffolds, it is important that we can look into the 3D scaffolds and to see that the fibroblasts have different features if they are in co-culture with tumor cells or not. I just run also a little through here. These are matrices where we know that can work even into 200, 300 micrometer depth and can see treated cells and compare with non-treated cells. And this is a possibility to also look into the quality of tissue that has been generated from autologous cells. So we can look into the quality of the cells after they have been in culture from the patient to make sure there's no contamination. And we can also look then into the cells in the scaffold to see how they have been grown, whether they are okay, whether they have been decayed, and this also goes up to 300 micrometers in depth. And so finally, it's a possibility for quality control of the final tissue graft before it goes to the patient, which is much quicker than the normal processes you need for FACS or MAX analysis or for DNA count. So last but not least, it's also possible to work in cells in this kind of microvals and follow up how agents treat or work on them. So you can take cancer cells and look how cancer agents work on them. In this case, we worked on apoptosis. So that is my last slide. We think that it is very important to work with small amount of cells to give an information without killing the cells, keep them happy. That's our motto, happy cells, healthy people. We have a lab where you can bring your cells and work with them to see how Raman analysis works for you. And we are located in Munich, close to Munich at Lake Starnberg. And thanks very much for your attention and for all who have contributed to this work already.